So welcome to Chapter 5, everybody. So in this week, we're going to be covering the plasma membrane diffusion osmosis. So this is actually one of my favorite chapters to cover since it actually integrates a lot of the content we've discussed before. So a lot of the biochemistry and organelles and structures and things like that are going to come up again. So that's one of the reasons why I really like lecturing on this chapter. And so a little bit different from previous lectures, we're actually going to start with questions first. And I won't be going over the answers until a little bit later in the lecture. So on your Blackboard page, just answer these questions, but of course don't submit your quiz just yet, since later in the lecture we're going to go over the actual answers. So here's question 1. So which of these best demonstrates the orientation of phospholipids in water? A, B, or C? Here's question 2. If you didn't get enough time to answer that question in the previous slide, of course you can go back and pause the video. So here's question 2. So which of these best demonstrates the orientation of phospholipids now in oil instead of water? So again, you have 3 possible answers, A, B, and C. So take a second to answer those, and I'll move on to the next slide in a couple of seconds. Here's question 3. So below is a molecule of detergent, also known as soap. Which boxed region is polar? A, B, or C? Here's question 4. So the following protein is embedded in the plasma membrane. The region pointed at or indicated is either A, hydrophilic, B, hydrophobic, C, neither, or D, both. And here's the last question from this set, so question number 5. Which of these leads to a more fluid membrane? A, B, C, or D? Okay, so just as I said, now we're actually going to go over the answers. So hopefully that was a good little pre-quiz for you to sort of get your mind thinking about the content for today. So first we're going to go over questions 1 and 2. So just to remind you what those questions were. Question 1 was asking you what the orientation of phospholipids is in water. And question 2 was asking about the orientation of phospholipids in oil. So just to remind you what phospholipids are and what they make up, here we have the plasma membrane, also sometimes known as the cell membrane. So this is what we talked about in the last lecture, actually. So here we have our phospholipids. So they are the major component of the plasma membrane. We also have proteins as well, but here we're really focusing on the phospholipids. So remember, phospholipids have a hydrophobic region. Hydrophobic, remember, means water-fearing. So this is something we talked about in the biochemistry lecture. And they also have a hydrophilic region. So water-loving. So if you imagine if I had a phospholipid right here, it has a head, which is hydrophilic. And then it has 2 hydrophobic tails. So this information is going to become useful for the next term I'm going to introduce to you guys. So this is something we briefly actually talked about in the biochemistry lecture. But remember that like interacts with like. So really what we mean here is that hydrophilic things like interacting with hydrophilic things, and hydrophobic things also like interacting with hydrophobic things. Another way you can put this is polar things like interacting with polar things, and nonpolar things like interact with nonpolar things. And that's exactly what it says right here. So polar interacts with polar. Nonpolar interacts with nonpolar. So if you imagine in the previous slide, if you go back, on the outside of the cell we have water. This is true for all of life, so all of life is surrounded by water, but also remember that a lot of the cytoplasm is also made up of water. So water is a major constituent both outside the cell and inside the cell. So here we have something that's polar and another substance that's polar on the inside. So this polar water is going to like interacting with the hydrophilic heads. It's going to dislike interacting with the hydrophobic tails. So you can imagine, if we go back to question number 1, imagine we have water on the outside of all of these. So you have water molecules surrounding all these phospholipids. So something like that. So you have water surrounding these phospholipids, and remember the phospholipid has a hydrophilic region and a hydrophobic region. So it's going to adopt an orientation that minimizes the amount of hydrophobic region interacting with water. So as you can see here, we have this hydrophobic region interacting with water. That's not good, it's bad. Here we have a lot of hydrophobic regions interacting with water again. And so here, really the best orientation is where you have the hydrophobic tails on the inside, and this minimizes the amount of water that's coming into contact with the hydrophobic tails. And if you look again, it's a nice textbook picture. Again, you can see the hydrophobic tails, just like in that question we had earlier, are facing inwards. So the hydrophilic heads are interacting with water, and the hydrophobic tails are interacting with themselves. So this is minimizing the amount of hydrophobic molecules or atoms or elements, however you want to put it, that are interacting with water. And so this is actually the most energetically stable arrangement of phospholipids. So again, we can go to the second concept here for the second question I asked. So nonpolar things like interact with nonpolar things. So this time we're looking at the arrangement of phospholipids in oil. So oil, if you remember, is a lipid, so it's nonpolar. So essentially, this is just a flipped version of the last question. So as opposed to water being the molecule surrounding the phospholipids, we have oil instead. Whoops. So here, essentially, the phospholipids are going to flip. So they're going to have as much of the hydrophobic regions exposed to oil as possible. And it's going to minimize the amount of hydrophilic heads that are in contact with that oil. So essentially, the phospholipids have flipped, so now they have their tail sticking outwards and their head inwards. So we've maximized the amount of the fatty acid tails of the phospholipids interacting with oil and minimized the amount of the hydrophilic heads interacting with the oil. And again, this is the idea of like interacts with like, polar things like interacting with polar things, nonpolar things like interacting with nonpolar things. So here's question 3. So below we had a molecule, in this case detergent, and I want you to identify the region that is polar. And so this goes all the way back actually to chapter 2 when we talked about basic chemistry. So polarity is all about electronegativity. If you remember, the most electronegative element is fluorine. And then as you go left or down the table, elements become less electronegative. So here we need to look at the types of bonds that are present. So here we have sulfur, oxygen. We have an actual ionic bond that's part of this molecule, which is very interesting. And then here we just have a bunch of carbons and hydrogens. So essentially each bend, if you remember, we talked a little bit about this, essentially shorthand for carbon and hydrogen. A little bit messy, but I think you guys get the picture. So essentially each bend in the line represents a carbon molecule. So for this detergent, we actually have a long stretch of carbons and hydrogens. So in regards to electronegativity, carbon and hydrogen are about equally electronegative. So essentially all these bonds right here are nonpolar, nonpolar, nonpolar. So we essentially have a long stretch of nonpolar bonds here. So this region right here, A and B, is really nonpolar. However, the region on the right is polar. And again, it's because of these oxygen-sulfur bonds. Also, you can think of the ionic bond almost being like a very, very strong polar bond in a way. So really, region C is the polar region of this molecule. Regions A and B are nonpolar. And so this brings up a concept we briefly touched on in an earlier lecture, and that's the concept of amphipathic molecules. So these are molecules with both a hydrophilic and a hydrophobic region, or put another way, a polar and a nonpolar region. So this detergent again has a nonpolar tail and a polar sort of head region. And here's the answer for that question just written out. So again, this region right here is nonpolar, and this region right here is polar. So a good way to figure out if a molecule is polar or not is to look for these major elements. So sulfur, oxygen, fluorine, nitrogen, and phosphorus. So if you see lots of those molecules, generally that molecule is going to be polar. If you see long stretches of carbons and hydrogens, though, as we see with the tail of this molecule, that usually points to it being nonpolar. Exactly what we see here. So your long stretches of carbons. So this region of the molecule is nonpolar. And so we've heard of an amphipathic molecule before. We talked a little bit about this again in biochemistry. But phospholipids themselves are amphipathic molecules. So here we have a phosphorus, nitrogen, lots of oxygen here. So the sort of head group is hydrophilic or polar, and the tails, again, lots of carbons, are hydrophobic. So essentially the head of the phospholipid molecule is hydrophilic or polar, and the tails are hydrophobic or nonpolar. So this again relates back to some of the content we covered a little bit earlier in this lecture. So here's question 4. So the following protein is embedded in the plasma membrane. The region indicated is A, hydrophilic, B, hydrophobic, C, neither, or D, both. And to answer this question, we need to go back to like interacts with like. And you also have to think back to the biochemistry lecture. So remember, we have polar or hydrophilic amino acids, but we also have nonpolar or hydrophobic amino acids. So depending on the amino acid sequence of that protein, certain regions can be hydrophobic and certain regions can be hydrophilic. We also have to imagine that this is a living cell, so there's lots and lots of water on the outside, water there, water on the inside as well, on the cytoplasm of the cell. So really this red region is interacting with water. So we'd expect like interacts with like, so we'd expect it to be hydrophilic. So it's going to be interacting with water. And again, that region of the protein would have lots and lots of polar or hydrophilic amino acids that make it up. And here's the last question, question 5. So which one of these leads to a more fluid membrane? A, B, C, or D? So to answer this question, we have to go over a couple of concepts first. And the first concept is saturated versus unsaturated fats. So depending on how closely you read chapter 3, you may already know this information. However, it wasn't something we actually covered in the lecture video, so I'm going to review those concepts now, just so everybody's on the same page. So on the left-hand side of this picture, we actually have a saturated fat. On the right-hand side, we have an unsaturated fat. And we're going to look at these triglyceride molecules right here. So remember, a triglyceride has two major parts. It has a glycerol, and then it has 3 fatty acid tails. That's the same for both saturated and unsaturated fats. The major difference between the two is the number of double bonds in the fatty acid tails. So looking at the zoomed in picture right here, we can see we have a carbon right here and a carbon right here, and they're linked together with a single bond. So they're sharing essentially 2 electrons. So again, relating back to a lecture we talked about a long time ago, this is chapter 2, basic chemistry. There are single bonds, 2 electrons shared in a covalent bond. Double bonds are sharing 4 electrons, and then triple bonds are sharing 6 electrons. So you can have different numbers of electrons shared in covalent bonds. So in the case of saturated fats, you have lots and lots of single bonds in the fatty acid tails, specifically single bonds between the carbons. In an unsaturated fat, you have lots and lots of double bonds in the fatty acid tails. So again, looking at this sort of zoomed in picture right here, you can see there are carbons linked together with double bonds. So what exactly does this all mean together? So if you look closely, you can see single bonds create molecules which are fairly straight. So they're just straight tails. So you can see they layer together very nicely, and they stack very compactly. If you look at the unsaturated fat though, when you have double bonds, this actually creates a kink in the molecule. Here's another double bond right here, creates another kink. Here we have a couple double bonds. So you can see the unsaturated fat with all its double bonds is actually less compact than the saturated fat. And this actually relates to membrane fluidity, which we'll talk about in the next slide. But when you think of saturated versus unsaturated fats, you actually think of food. So saturated fats are things like butters. You have butter. So butter is made up of a lot of saturated fats. So you can imagine all these triglycerides stacking on top of each other. They're very, very compact, and that makes butter a solid at room temperature. An unsaturated fat is something like oil. So here we have probably olive oil in a bottle. So you have lots and lots of unsaturated fats. And again, it has lots of double bonds, and that creates these kinks. So these fatty acids, and more specifically, these triglycerides can't stack together. So they're much less compact and they're all moving around, and this makes oil a liquid at room temperature. So really, saturated versus unsaturated fats is all about the compactness of triglycerides. Okay, so how does this all relate to membrane fluidity? So you can imagine just like we have saturated versus unsaturated triglycerides, we can also have saturated versus unsaturated phospholipids. So just like in saturated triglycerides, saturated phospholipids are very compact. Unsaturated phospholipids, though, are very spread out. So just like in a saturated triglyceride, like butter, that's very compact, that's going to be more solid. An unsaturated phospholipid, as opposed to a saturated phospholipid, is going to be more liquid. So again, butter versus oil. Exactly what it says here. So double bonds between carbons increases membrane fluidity. Single bonds decrease fluidity. So the second thing we need to cover to answer the question is the fact that cholesterol can actually integrate itself into the plasma membrane. So if you remember back to biochemistry, cholesterol is a lipid molecule which makes it nonpolar. If you also remember back to that lecture, it has this 4-ring structure right here. So cholesterol can actually sort of wedge itself between phospholipid molecules and actually integrate itself into the plasma membrane. So we're going to actually have a variety of functions depending on the temperature that surrounds the cell. So at low temperatures, it actually increases fluidity. So low temperatures increase fluidity. And the flip side of that, at high temperatures, it actually decreases fluidity. So if you take this together, essentially cholesterol maintains membrane fluidity. So at low temperatures, when these phospholipids are sort of more compact, it's going to help space them out. At high temperatures, when the phospholipids are sort of spread out, it's going to help compact them back together. So really, the main function of cholesterol is to maintain membrane fluidity. Okay, so now we've covered all of that, let's go back to the actual question. So here you have 4 different possibilities for question number 5. So the first one is a decrease in the number of double bonds in the fatty acid chain will make the membrane more fluid. So remember here, double bonds means more fluidity. So an increase in double bonds means an increase in fluidity. Here we're doing the exact opposite of that. So we're making more single bonds. So if we go back to the next slide, actually, you can see that single bonds are more characteristic of saturated fats or saturated fatty acids like butter. So A is not the correct answer, since if we decrease the number of double bonds, we're making the lipid bilayer or plasma membrane less fluid. Let's look at B. So an increase in the number of double bonds in the fatty acid chain will make the membrane more fluid. So that follows this. So we're increasing the number of double bonds, and that makes the membrane more fluid. So we're going to put a check mark here, since it looks like that's the best answer. However, just to be sure, we're going to go over C and D to make sure those answers are incorrect. So C, a decrease in cholesterol at low temperatures will increase membrane fluidity. So remember, cholesterol is all about maintaining membrane fluidity. So you actually want an increase in cholesterol at low temperatures. So if you increase that amount of cholesterol, it's going to make the membrane more fluid at low temperatures. Let's look at D, an increase in cholesterol at high temperatures. So remember, cholesterol is all about maintaining membrane fluidity. So at high temperatures, cholesterol actually decreases membrane fluidity. Since those phospholipids are spreading out, cholesterol is actually making them more compact. So by increasing cholesterol at high temperatures, you're actually making the membrane less fluid. So again, if this was going to be a correct answer, you want to decrease the amount of cholesterol. So let's leave this again with B as being the best answer here. So an increase in the number of double bonds in the fatty acid chains of the phospholipids will lead to a more fluid membrane. And so the next thing I would like to actually cover is the actual nature of the lipid bilayer or plasma membrane. So I think a lot of the pictures I've shown really can't do it justice. So I'm actually going to show you guys a quick animation that's going to go over a lot of the content we just covered, as well as show you what the actual plasma membrane looks like in a living cell.
Between the living machinery of the inner cell and the harsh conditions of the outside world stands the cell's plasma membrane. As crucial as this barrier is, it's surprisingly flexible. Push it and watch it move. Poke hard enough and it might break and begin to regroup. The lipid molecules of the membrane naturally assemble in a double layer because their tails repel water as their heads attract it. Throw in some cholesterol and a few carbohydrates, and you have the basic structure of a plasma membrane. Within these lipid molecules, we also find different proteins which do various things for the cell. For instance, they receive signals from the world outside, or they transport nutrients and waste. So nature composes the membrane with a combination or mosaic of different lipids, carbohydrates, and proteins. And these molecules are not stationary. They constantly move within the structure, fluidly changing their positions. The survival of all life rests on this veil of material. A supple membrane just 2 molecules thick.
So hopefully that gives you a pretty good idea of what the plasma membrane actually looks like. So again, it's not something that's static and not moving. It's actually very flexible, and you have proteins and other things moving around all the time. So don't think of it as just some non-moving thing. Really, it's been compared to sort of salad oil in thickness or viscosity. So you can sort of imagine salad oil. You have molecules sort of moving around constantly, and it's very liquidish. So the next thing we're going to talk about is diffusion, osmosis, and active transport. As a video sort of alluded to, there are actually proteins embedded in the plasma membrane. And we're going to see that some of those proteins actually play a huge role in a couple of these functions. And as sort of an overview, I have this slide right here. So we're going to talk about diffusion, osmosis, and active transport. This slide right here talks specifically about diffusion. So there are really two types of diffusion, actually. So you have just plain diffusion, and you have facilitated diffusion. We're going to talk a little bit about the difference between these two in a later slide. You can actually group diffusion and facilitated diffusion under the term passive transport, and that's because both of these require no energy. So remember, we have that energy molecule ATP. That's the energy molecule the mitochondria makes. So here, no ATP is used for these processes. For active transport, though, as the name sort of alludes to, this requires ATP. So it requires energy. And now we're going to go over a new concept, and that's that things move down their concentration gradient. And by things, I actually mean solutes. So I'll talk a little bit more about what this means in lab, but you can think of, if you're making like Kool-Aid or something, the solute is the Kool-Aid packet, and then the solvent would be water. So the solute is what dissolves in the solvent. So in this case, the Kool-Aid packet is the solute, and the solvent is water. So the solvent is what dissolves the solute, and the solute is what dissolves in the solvent. And we'll use this example of a Kool-Aid packet again to explain this term right here, or this concept, that solutes move down their concentration gradient. So you can imagine if you're making Kool-Aid, once you add that packet of Kool-Aid, so the flavoring, the sugar, and the food coloring, at the interface between the two, so the interface between the solvent and the solute, you have an area of high concentration. So the Kool-Aid is very concentrated at that interface. As time goes on, though, you'll see that the Kool-Aid will actually spread out. So you'll see that the food coloring will actually spread, and it will evenly distribute itself across all of the solvent, or essentially across all of the pitcher or bottle of water you have. That's, of course, assuming that you don't mix the Kool-Aid together with the water and artificially speed up the process. And that's the concept of solutes moving down their concentration gradient. So it's exactly what I show in this diagram on the bottom. We have an area of high concentration on the right, and over time, so just imagine we left the solvent and solute just sit for a while, you're going to see the solute slowly diffuse or spread out over time. So again, it's going to go from the area of high concentration to the area of low concentration. In this case, from the right to the left. It's going to look like this over time. So again, you're going to have an even distribution of that molecule over time. And so this all taken together is known as diffusion. So going from the area of high concentration to the area of low concentration. So to sort of drive that point home, here's a question for you. So which way would the following molecules diffuse? A left, B right, or C there would be no change? So before I answer that question, let's go over a couple of the key terms we talked about in the last slide. So the solute in this case is little green circles here. So that's what's dissolved in the solvent. In this case, the solvent is this blue stuff right here, which is likely water. So again, the solute dissolves in the solvent, and the solvent is what dissolves the solute. So here we're going to see the movement of the green solute molecules from the area of high concentration to the area of low concentration. So again, we have low concentration on the right, we have high on the left. So we're going to see the molecules move from left to right. And in this case, the correct answer is B. So we're going to see the molecules move to the right. And so we've talked about the plasma membrane today. And this concept of diffusion relates heavily to the idea of selective permeability. So remember, that's one of the major functions of the plasma membrane. It's being selectively permeable, so allowing some substances to pass through and others not. So just like we saw diffusion in the artificial examples in the previous slides, we're going to see diffusion happening in the cell itself. And that's demonstrated by this figure on the bottom. So you can see you have a high concentration of blue solute molecules on the outside of the cell, and over time, you can see those blue molecules diffuse inside. So they pass the plasma membrane and they diffuse inside the cell. So here's another question for you. This is question 7. So what would happen if the membrane was not permeable to the blue molecule? Would it either A, it would diffuse outside, B, it would diffuse inside, or C, there would be no change? So what would happen if this membrane was not permeable to this blue molecule? So even though we have a high concentration outside the cell and a low concentration inside the cell, we're still not going to see any diffusion happening here. Again, the membrane is not permeable to this molecule. Since it's not permeable, the molecule is not able to pass through and diffuse inside the cell. So the correct answer here is C. There would be no change. Although if you can imagine if this was an infinite space, these blue molecules would diffuse outward into that space. But here that's not a possible answer. So A and B is, would it diffuse outside the cell or would it diffuse inside the cell? And that leaves us with C, no change. Again, the plasma membrane is not permeable to the blue molecule, so we're not going to see any diffusion happening. So really what we've been talking about is passive diffusion. So that's the more correct technical term for what we've been discussing. So diffusion is just the movement of solutes in a solution, but passive diffusion is really relating it to the actual cellular function. So again, it uses the term passive, and that relates to the fact that it requires no energy input by the cell. So no ATP is used, so this is a passive function. And here I just have another picture showing just what we've seen before in the last examples. So here we have the plasma membrane. We have some blue hexagonal solutes, and over time, they go from the area of high concentration to the area of low concentration. So they're moving inward. And eventually, at the very end of this diagram, you'll see that there's an equal amount of solute molecules outside the cell as well as inside the cell. So again, this is passive diffusion. So here's a question for you to ponder. So what region of the phospholipid or lipid bilayer takes up the most space? And if you want to, use the diagram below to help you formulate your answer. So now that you've taken some time to do that, hopefully you came up with the hydrophobic region taking up the most space. And that's absolutely correct. So as you can imagine, a phospholipid has a very long tail and a very short or tiny head region. So actually the hydrophobic region takes up the most space. So now let's think about that. So remember that like interacts with like. So this is a concept we've talked about a few times now. So what do you think would pass through the plasma membrane most easily? So if you came up with nonpolar hydrophobic molecules, you're absolutely correct. So remember, we have quite a bit of space that those molecules need to travel through. So they travel mostly through a hydrophobic space. So in general, nonpolar molecules or hydrophobic molecules can pass through the plasma membrane more easily than polar or hydrophilic molecules. And again, that's because there's so much hydrophobic space in between the plasma membrane or the lipid bilayer that those molecules need to cross compared to the hydrophilic regions. So here's question 8 for Blackboard. So which of the following do you think would diffuse most easily across a plasma membrane? A or B? So here you need to figure out first which of these molecules is polar. And the best way to figure that out is to know what these molecules are. So here we have a carbohydrate. Carbohydrates are always polar, so they're hydrophilic. On the right, we have our 3 tails and a glycerol group, so this is a triglyceride. So this is nonpolar. And so again, the hydrophobic tails of the phospholipids are so long compared to the hydrophilic heads. So in this case, B is the correct answer. A triglyceride passed through the plasma membrane much more easily than a carbohydrate. Again, because the triglyceride is nonpolar or hydrophobic. So next, we have a similar question. So question 9. So which of these would diffuse most easily across the plasma membrane? A or B? And so even though we have a polar molecule here, so even though water is polar, it's much, much smaller than the molecule on the right. The correct answer here is A. So in general, small molecules pass through the plasma membrane more easily. So you can imagine something small fitting through the plasma membrane is much easier than something huge, since something big is going to have a harder time squeezing through the phospholipid bilayer. So in general, for passive diffusion, small nonpolar molecules diffuse the best. So again, things like triglycerides, water, even though water is polar, it's still very small, oxygen, carbon dioxide, so common gases. So small nonpolar things diffuse the best. And again, that's because it has to squeeze through the lipid bilayer, and it's also because it has to interact with the hydrophobic tails. So something that's hydrophilic or polar is not going to be very happy trying to squeeze past those hydrophobic tails. It's not going to like interacting with them. So small nonpolar things diffuse the best. So next we're going to talk about a very important term called osmosis. This doesn't really fit in with passive diffusion, but it's sort of similar in a way. So again, we've been talking about passive diffusion, and that's movement of solutes, or essentially dissolved molecules. Osmosis is the exact opposite of that. So as opposed to the movement of solute molecules, here we're moving solvent molecules. And in most cases, that solvent is going to be water. So osmosis is all about the movement of water as opposed to solutes. And here water goes from the area of low solute concentration to the area of high solute concentration. So water moves from the low solute concentration to high solute concentration. On this slide, I actually have a picture that shows that. So let's orient you to this picture really quick. So here we have a beaker. It's full of sugar and water. So here, sugar is a solute, and water is a solvent. In this beaker, we have a membrane, and it says semi-permeable. So it's important to note here that this membrane is not permeable to sugar. However, it is permeable to water. So if it was permeable to sugar, we would just have regular diffusion happening here. But because it's not permeable to sugar, we have osmosis happening. So on the right-hand side, we have a high concentration of solutes. On the left-hand side, we have a low concentration of solutes. And again, this membrane is permeable to water, but not sugar. So you're not going to have diffusion, you're going to have osmosis. And osmosis happens from the area of low solute concentration to the area of high solute concentration. So that is, water is going to move from the low solute concentration area to the high solute concentration area. And again, if you just mark which side is high and low, this is a straightforward question. So then it's going to move from left to right. Again, from the area of low solute concentration to the area of high solute concentration. That's exactly what we see. So after some time has passed, we can see that there's now much more water or solvent on the right-hand side of this beaker compared to the left-hand side. And so this process of osmosis is incredibly important for life. So before we go into the figure on the right, we're going to talk about a couple more terms. And they're hypotonic, isotonic, and hypertonic. So all of these terms relate to the salt or solute concentration compared to the inside of the cell. So for something like a hypotonic solution, you can think of the word hypo meaning low or below. So it has a low solute or salt concentration outside the cell compared to inside. Isotonic, iso means same. So same salt concentration or solute concentration outside the cell as inside. And then hypertonic means high salt or high solute outside the cell compared to inside. So here let's look on the figure on the right. So on the first column, we have a hypotonic solution. So again, low solute on the outside, high solute on the inside. And here we're going to have osmosis happening, not diffusion. So we go from the area of low solute concentration to the area of high solute concentration. In this case, water comes in to the cell, and this will actually burst an animal cell. So here we actually have red blood cells, so the cell that carries oxygen in the blood. You can see that it bursts when you expose it to a hypotonic solution. So this is one of the reasons why when they inject you with a solution or a vaccine or give you an IV fluid, they always use an isotonic solution. If they just use tap water or something like that, you're going to have a hypotonic solution that's going to cause all your red blood cells to burst. It's going to make you very sick, and you're going to become anemic. So this is why they have to use actually an isotonic solution when they inject you, because you don't want those red blood cells to burst because you're injecting a hypotonic solution. And so the technical term for when this happens, when a cell bursts, is lysed or lysis. So you can think of hydrolysis, where you break things down with water. Lysis or lysed cells have been broken open. And so looking at a plant cell on the bottom part of this column, this is actually normal for a plant cell. So because the plant cell has a cell wall, so remember outside the cell membrane, they have that cell wall protecting the cell. This is actually okay for a plant cell. So they're happy being in a hypotonic solution, which is why you can give your plant tap water and they're okay with that. And the technical term for this is turgid, so this is normal for a plant cell. So looking at the second column, isotonic solution, so essentially we have equal salt or solute outside the cell as inside. So here, the red blood cell is happy. It has this nice biconcave or disc shape. So that's, again, why we inject isotonic solutions via IV or vaccines or whatever you're being injected with, since you don't want to burst those red blood cells. For a plant cell, though, they're actually flaccid in isotonic solution. So if you give them salt water or something like that, your plant's not going to be very happy. And last column, we have a hypertonic solution. So this is where you have high salt outside, low salt inside. Again, we're going from the area of low solute concentration to the area of high solute concentration. So water leaves the cell. So in a hypertonic solution, this will cause a red blood cell to become shriveled. And in a plant cell, the technical term there is plasmolyzed. So you can see the cell wall is actually intact, but the cell itself has actually shrunken. So here's the actual cell membrane, which you can see has shrunken down because all the water is leaving the cell. So depending on the type of cell you're talking about, for a plant cell, they like being in hypotonic solutions, and a red blood cell likes being in isotonic solutions. So as you can see, this process of osmosis is very important for cells, especially for human health and also for agriculture and plant biology. So next we're going to talk about facilitated diffusion. So just as the name sort of alludes to, facilitated diffusion requires a protein or help, essentially, for diffusion of molecules. So when something cannot passively diffuse, a protein is needed. So these are things that may be like ions, large molecules, things like that. So remember, passive diffusion, small nonpolar molecules passively diffuse very well. For things like ions, polar molecules, large molecules, they may require a protein to help them. Oftentimes, these proteins are called channel proteins. So right here and here are two examples of channel proteins. So you can imagine these proteins are creating a channel for the ions or large molecules or what have you to pass through the plasma membrane. Just like passive diffusion, though, molecules move from the area of high concentration to the area of low concentration. So here for like these orange triangles, they go from high to low. For these blue circle molecules, they go from high to low. So in facilitated diffusion, you still have molecules or solutes traveling from high to low concentration. The major difference here is it requires the use of a protein. And that's because these molecules would not be able to diffuse passively because they're not able to pass through the plasma membrane. It's also important to note here that this requires no energy, so no AGP is used by these channel proteins to move these molecules. And lastly, we have active transport. So active transport requires energy. The reason why it requires energy is because unlike facilitated and passive diffusion, active transport allows molecules to go against their concentration gradient. So it allows them to go from an area of low concentration to area of high concentration, which is exactly what we see in this picture here. So we have two green molecules and a bunch of green molecules on the right. So we go from the area of low to the area of high. So essentially we're going from the area of low solute concentration to the area of high solute concentration, which is backwards from passive diffusion and facilitated diffusion. And because you're going from low to high, this costs energy. So here we actually have an active transport protein. And you can see it's using AGP to drive this process. So active transport requires a protein as well as energy to have it occur. And so let's go back to the summary slide. So we've talked about diffusion, also known as passive diffusion. We have facilitated diffusion, which requires a protein. And these can be lumped together under the term passive transport. And lastly, we talked about active transport. It also requires a protein, but most importantly, it requires energy. For both diffusion and facilitated diffusion, again, we go from the area of high solute concentration to the area of low solute concentration for both. Active transport is different, though. It goes from the area of low solute concentration to the area of high solute concentration. We also learned about osmosis, which is different from these three processes. That's about the movement of solvents. And in life, 99.99% of the time, that solvent is going to be water. And so the absolutely last process we're going to talk about are exo and endocytosis. So exo, of course, means outside. Endo means inside. So I like to think of exo and endocytosis almost being like eating and vomiting. So exocytosis is more like vomiting, and endocytosis is more like eating. And if you look at the figure on the bottom, we can see why this is true. So imagine a cell is going to ingest some food. So we have some food particle right here. In endocytosis, essentially you have an infolding of the plasma membrane. So to the outside of the cell, there's an infolding of the plasma membrane. It's almost creating like a pocket. So it has an infolding, and eventually these two sort of ends are going to come together, and you have a food vacuole formed. So essentially by the pinching together and sort of this in-pocketing of the plasma membrane, the cells actually ingested some food. So that's endocytosis. So bringing something inside the cell, which is why it gets the endo prefix. And exocytosis, just as I said, is more like vomiting. So it's almost the exact opposite of that process. So here you may have some digested material. You have a vesicle formed, and that vesicle then fuses with the plasma membrane, and that food is spit out of the cell. So essentially through this process, the cell has expelled food or some other sort of particle into the outside environment. So this is the process of exocytosis or endocytosis. So this is moving large quantities of molecules or food or what have you inside and outside the cell. So you can think of passive diffusion, facilitated diffusion, active transport being about small movements of molecules. Here we're moving very large substances across the plasma membrane. And here's a table to help you review. So here we have the three major types of transport: passive diffusion, facilitated diffusion, active transport, whether they require proteins, whether or not they move down or up the concentration gradient, and whether they require energy. So a helpful hint for studying, I would highly recommend is that you blank out this table, essentially make this part blank, and then see if you can fill it in from memory. So see if you understand all of these processes.